go.
Okay, hi everybody. My name is Lauren. This is Henry, Casey, Emma, and JP. So what if I said you had a debilitating disease with a shortened lifespan and a pretty low quality of life? Would you participate in a clinical trial if it meant that you could cure the disease? Show of hands. Okay, so what if I said that this clinical trial might give you leukemia? Would you still do it? Yeah? Okay, so that brings us to Sam. Sam has X-linked severe combined immune deficiency, or SCID. This mutation causes a defect on his interleukin receptors, giving him immature B and T cells. This mutation forces him to live in a sterile environment because of his lack of immune system. Originally, Sam looked at bone marrow transplants, but these are not 100% effective for him. So where does he go from here? Well, Sam looks to gene therapy, of course. Gene therapy takes a synthesized functional copy of a gene, inserts it into a defective cell, and hopes that it complements and gives it a function. There are two different types of this, ex vivo and in vivo. Ex vivo takes out some of your mutated cells, puts the virus in, and then puts it back in. In vivo just inserts the copy right in. So who can benefit? It's important to note that SCID is not the only people that can benefit from this. Anybody with a rare recessive inherited mutation can benefit from gene therapy, as well as more complex diseases like retinal degeneration and HIV. So different modes of treatment. Originally, we looked at retroviruses, which insert into the host genome, and adenoviruses, which do not insert into the genome, but instead exist as an episome. Today, we're looking at more adeno-associated viruses, which again don't insert into the genome, and lentiviruses, which do insert, because these last two have less adverse effects than the first two. And the hope is that we can eliminate these adverse effects to create a more across-the-board therapy. So back to Sam. He, again, like I said, tried this bone marrow transplant. It did not work, so his next resort was a clinical trial with Alan Fisher out of Paris. The hope of this trial was to insert a functional copy of his gene to replace his non-functional copy to give him a functional immune system and mature T cells. The problem with this is that there are two different types of SCID, one that focuses on chromosome 20 with the ADA gene and one that focuses on the X chromosome. So did Sam get a functional immune system? Yes, which is awesome. So he had a functional immune system, his T cells were working. Unfortunately, 5 of the 20 patients in this trial developed a rare form of leukemia that doctors had never seen before. So what does this mean? Luckily, 4 years after the trial finished, 4 of the 5 leukemia patients were in remission and still doing very well. This led Alan Fisher, the private investigator on the study, to think what went wrong. Well, he looked at his retroviral vector and saw that it inserted too close to the proto-oncogene LMO2, which caused a rapid proliferation of his T cells, obviously causing cancer. Fisher decided that he had no way of controlling where this vector was being inserted and what the effects were going to be. So instead, he decided just to cancel his study because he couldn't control the outcomes. So some other failures, unfortunately. In 1999, Jesse Gelsinger was using gene therapy to correct a rare form of liver disease with an adenovirus vector. Unfortunately, a few days after receiving the vector, he suffered a massive immune response, which actually killed him. So this massive immune response, these cancerous effects, really did nothing to support gene therapy, but instead hindered the research that was being done on it. But we, of course, have some successes. The first was in 1990, when an autosomal form of SCID was corrected by inserting a functional copy of this ADA gene, giving these patients an immune system. Another one was in the UK, where 14 of 16 patients who experienced SCID received or had a functional immune system 9 years after the trial was finished. Other successes have been in retinal degeneration, which is more of a complex disease than some of these immune deficiencies. So retinal degeneration uses an AAV, adeno-associated viral vector, that targets these localized and infrequently dividing retinal cells. Because it doesn't integrate into the host genome, there's no cause of cancer for this type of vector. And because these cells do not divide frequently, treatment does not have to happen that often. And patients in this trial experienced a 200 to 60,000-fold increase in light sensitivity. Another clinical trial is right down the road at Boston Children's Hospital, which uses AAV vectors to target the TMC1 gene, which causes deafness in mice and in some humans. So this TMC1 gene targets proteins that turn sound waves into electrical signals. And this trial saw great sound sensitivity in all mice, and their hope is that they can now transfer this into human subjects. So the future of gene therapy. We've obviously talked a lot about these AAV vectors, but the future is definitely in this lentivirus. Lentivirus is a promising retrovirus that has a larger carrying capacity, which means it can take up these self-inactivating promoter sequences, which stop random integration. They also can take up suicide genes, which initiate cell apoptosis if the functional gene gets too close to this proto-oncogene, thus eliminating the effects for cancer. So obviously we would have some ethical issues with gene therapy, one of them being that we don't really know the effects of gene therapy and what it has for the future of patients. So is it worth putting people under the knife, so to speak, if we don't know what's going to happen to them? Another big ethical concern is with germline therapy. Can we correct people's germline cells if it means changing generations worth of diseases, and what does that mean for the generations? So just to wrap it all together, why hasn't gene therapy been delivered to the community? Mostly because of these early setbacks. Cancer, this immune response, people are afraid of it and don't want to go full force into it. However, because of research onto the AAV vector and the lentivirus vector, we're learning how to control vector insertion, thus eliminating some of these really hardcore risky effects. So now Casey's going to give you a few of our propositions on where we want